Okay, welcome to our video on enzymes. This links straight on from the video on proteins, so I'm hoping that you've made sure that you've watched that one and you're up to speed with your proteins before you start onto this one. Okay, here are our outcomes. We should be able to state all of the enzymes found in the digestive system, what those enzymes break down, so their substrates, and what products are formed. We also need to be able to explain how temperature and pH will affect enzyme structure and how that will have an effect on the function of an enzyme. And then we need to talk about enzymes use in industry, because an awful lot of products that we use every day depend on enzymes and the industrial processes that go with that. Okay, here we go then. All right, here's the diagram of the digestive system. Now, you've talked about the digestive system earlier on in the course when we looked at the stomach, and we looked at that in detail, and we looked at the 3 different types of tissue found in the stomach. We looked at the muscular tissue, the epithelial tissue, which is the surface layer, and the glandular tissue that produces the digestive juices. So this diagram is actually the best diagram that I could find, but it has an awful lot more label lines than we need. So we're not going to use all the label lines, and that's fine. So we'll just concentrate on the bits we need to know about. First of all, obviously, food is going in through the mouth, which breaks up food and mixes it with saliva. There's one enzyme in saliva, which is salivary amylase, and salivary amylase breaks down starch. So starch is the substrate, and the products that are formed are small sugar molecules. All of the food then travels down the esophagus and enters the stomach. Because the stomach is muscular, it can churn the food around and mix it with even more digestive juices. So we've got hydrochloric acid being added in the stomach, and that's mixed around with all of the food and the amylase that's come down with it, because your saliva obviously travels down your esophagus too and enters the stomach. And during that churning, there's another enzyme mixed in along with the hydrochloric acid, and that enzyme is a protease enzyme. Now, protease enzymes break down proteins, and they're going to form short chains of amino acids. So if you think back to the proteins with the long chain of beads, if you like, with the beads being amino acids, protease enzymes in the stomach break down those big long protein molecules into short chains of maybe just a few amino acids. The food then travels from the stomach into the small intestine, which is this coiled up area lower down here. Lots and lots of twisty-turny tubes. And the small intestine has 2 really vital functions. First of all, the rest of digestion is going to take place there. So we've had a bit of digestion of a carbohydrate, starch, in the mouth. We've had a bit of digestion of protein in the stomach. And we need to carry on digesting carbohydrates and also proteins. And we also need to digest fats, and all of that is taking place in the small intestine. So we've got 3 types of enzymes involved in the small intestine. We've got more carbohydrates, which are going to continue breaking down all those sugars into glucose molecules. We've also got protease enzymes, which are going to continue breaking down the short chains of amino acids into single amino acids. And we've also got lipase enzymes. Now, lipase is going to break down all the fats and oils, because the proper name for fats and oils is lipid. It's where the word liposuction comes from. So lipase is going to break down fat molecules, and the 2 products of fat digestion are fatty acids and a molecule called glycerol. So we're going to get fatty acids and glycerol. Now, all of those molecules that I've just mentioned, glucose, amino acids, fatty acids, and glycerol, all of those molecules are small enough to be able to then move across and be absorbed from inside the small intestine into the bloodstream. They need to enter the bloodstream so that we can use them in our bodies. And the only thing that's going to be left then in the small intestine, continuing into the large intestine, is all the waste molecules. And the waste molecules, stuff that we can't use, plus all the fibre, is going to then continue through the large intestine, into the rectum, and out through the anus. The only thing that happens in the large intestine is that all the extra water that has been added and that you've drunk is going to be removed and put back into your bloodstream again. so that water travels from the large intestine into the blood and leaves the waste a little bit drier. If that didn't happen, if your food and all the molecules in there get rushed through your digestive system too quickly, the water isn't reabsorbed and you end up with diarrhoea. So obviously it's very important that the large intestine, even though it doesn't do any digestion and there's no enzymes there, it's still very important that the extra water is taken out of that waste and put into your bloodstream. Okay, just a quick recap about catalysts. You've done about them in chemistry. You know they speed up chemical reactions. The products can form more quickly than if the reaction took place without catalysts. And enzymes are biological catalysts. So the only thing that we need to think about here is the fact that they are just catalysts, but that they're produced by living cells. So because they're found in cells, because they're made by cells and they work in cells, they are biological catalysts. Okay, this slide is just a quick recap on protein structure. You know, and I remember us building them in class, that we've built proteins out of amino acids. And we just need to briefly remember what type of foods have a high level of protein. So we're looking at meat, fish, eggs, nuts, seeds, things that we call pulses, which are like beans and lentils and chickpeas. Okay. Okay, some proteins have special shapes and their shape is really, really important for the job that they do. Now there's going to be chemical bonding holding that shape together. So you can see in this diagram that we've still got the same chain of amino acids, but now it's been folded into a particular pattern and there's little red bonds holding it together in particular places, and that's going to keep it in its shape. So if it depends so much on its shape, and obviously therefore depends very much on those bonds holding it in shape, if those red bonds were broken, what would happen to the protein? And obviously I'm sure you can see that it would just unravel and fall back into, a single string that wasn't in the correct shape. If it's not in the correct shape, it won't be able to carry out its job properly. And this is a massive problem for enzymes because this can happen to them. Right, in this slide we can see that these are much more basic looking diagrams, and this is how we represent enzyme structures. We usually see them as a sort of blobby shape but with a specific area that's got a very specialised little shape to it. So if you look at the substrate molecule, which is this turquoise molecule down at the bottom, which of the three enzymes is it going to fit with? Now, scientists usually describe enzymes fitting together with their substrates as a bit like a lock and a key fitting together. So if you think of the substrate as being the key, which lock will it fit? And you can see quite clearly from the diagram that it's going to fit into enzyme number 1, which is the yellow one. It won't fit into either of the other two, and this is why enzymes are very specific to a particular reaction. You won't find a protease breaking down a carbohydrate molecule. You won't find a lipase breaking down a protein molecule, and so on. They're very, very specific. They'll only carry out one reaction, and you'll need a different enzyme to do a different reaction with a different substrate. If we look at the specific part that's got this special shape, we can see that we call it the active site. So the active site of an enzyme is the part where the substrate fits, and it's the part with the really specialised shape. And it's very, very important that that shape doesn't alter, because obviously then the substrate won't fit into it and the reaction won't take place. So we need the substrate to be broken into its products, and if the shape of the active site has changed, the substrate can't be broken into its products, those products won't be made. And we can see from what we've got here that the bonds can get damaged by excess heating. So if the temperature rose and rose and rose, we'd get to a point where that special shape was destroyed and it's no longer going to fit with the substrate molecule. Here's an example of just that. So if we look at the top one, we'll see that the substrate and the enzyme are going to fit together perfectly, and we're going to end up with the turquoise model being broken into its products. Whereas with the lower molecule, there's no way that that substrate is going to fit the active site, because the active site has changed and the products will not be formed. That turquoise molecule will not change into anything else. It will stay as it is. It will remain as a substrate. It's also important that we understand how pH affects enzymes. pH can have a similar effect to high temperatures in that it can cause the active site to change. Now, the difference is with temperature, only very high temperatures will cause that damage to the active site. Whereas with pH, any pH that is too extreme for that particular enzyme will have that same damaging effect. So in the digestive system, it's quite interesting because you've got hydrochloric acid, which has of course got a very low pH, around about pH 2, and for the enzymes that you find in the stomach where the hydrochloric acid is released, that's fine. That's its pH that it wants to work at, and we call that the optimum pH. So pH 2 for the enzymes in the stomach is absolutely brilliant. It will work very fast at that pH. But if you take it to a pH a few steps away, say pH 7, pH 8 and above, then that is going to cause the active site to be destroyed. On the other hand, you go to the small intestine and the digestive juice there, bile, has got a very high pH, it's alkaline, and the enzymes that you find there, the carbohydrates, the lipase and the protease, they work best at an alkaline pH. So it depends on what the enzyme is, where it's found, what the conditions are like there. They will work best at a particular pH, and if you go too far from that, too extreme, then the enzyme active site will be destroyed. There's a really useful word we can use here, and we can say that the enzyme is denatured. And denaturing only occurs at high temperatures or extreme pHs. We also need to know how enzymes are used by scientists in industrial processes, and there's a whole variety of different things that they're used for. For instance, this worried looking baby is probably eating jarred baby food, and when they make baby food, they actually add protease enzymes into it so that the baby doesn't have to digest all of the proteins to amino acids, that it's partly done for them, so they only have to do some of the digestion. It means that there's a much bigger chance the baby can absorb those nutrients, and the more amino acids the baby gets into its bloodstream, the more it'll be able to grow, and obviously that's really, really important. So baby foods that are produced in the little jars and pouches are usually what we call pre-digested, which means that there's protease enzymes in there with the food, making it a little bit easier on the baby's digestive system, because young babies don't have a very well-developed digestive system. In addition, if you see the box of washing powder there and notice that it says bio, that means it's a biological washing powder. And biological washing powders have enzymes in them, and that helps to digest the stains off your clothes. You can also get non-biological, and you can just see the very, very top of the blue box in the front of the picture. That's a non-biological washing powder, because some people have allergies to the enzymes that are used, and that means that they can't use biological washing powders. So people with eczema were more likely to be using non-biological washing powders. Biological washing powders also have the advantage that you can then wash your clothes at a lower temperature to remove the stains, because enzymes work at relatively cool temperatures, and they'll still be able to do the work that you'd otherwise have to boil your clothes to get the stains out. It will do that at a much lower temperature. So that's saving you money on your electricity, because you're not having to heat the water in your washing machine to such a high temperature. And obviously that's better for the environment too. Lots of other things we'll talk about in class that they do. Converting starches into sugars, converting sugars into other sugars for various reasons. Some of those are used in slimming foods, and we'll have a little look at that in class. Okay, if you go back and have a little look at the very start of the video with the outcomes, you should be okay with what we've been doing so far, and we'll continue it in class.